Hey, we are going for lesson 2.2 in microbiomes, beginning a case study of patient 23. Interesting. Hey, last lesson, one of the things that we did was we read about the human microbiome and some of the things going on inside of humans. We're going to revisit that article today. We're going to watch a video today. We're going to write down some answers to some questions today. And we're going to start this case study of patient 23. We'll talk about more of what a case study is in just a moment. But first, before we do anything else, let's go ahead and let's do our warm-up. There's our warm-up slide. And here it is. That's what it looks like on our slides. Here's what it looks like. Oh, wait, one second. It's over here. Here's what it looks like in your Amplify lesson. Boom, there it is. And here's your warm-up. After reading the Human Microbiome article, you learned that there are trillions of bacteria in the human microbiome. Which of these statements do you agree with most right now? Read through these statements. Decide which one you agree with most, click it. And then what other interesting things did you learn from reading the Human Microbiome article? So go ahead, answer those questions. Make sure you click hand in. I'm going to give you a second to do that and then come back to me. So what'd you say? Did you say they were disgusting? Did you say, no, they're great, I love them, they're my best friends? Did you say, I really don't know, Mr. Reagan, I'm still waiting for you to tell me if they're good or whatever. Just feed me information. Thank you. Okay. Well, I won't yet. Maybe eventually. Let's keep moving on. So, here we go. Activity 2, message from the Microbiome Research Institute. So if you're in Amplify, you will click number 2 right there. And look, a message from the Microbiome Research Inst. And then ellipses. I guess they couldn't pay to finish writing that. That's fine. Let's discuss your questions about the Human Microbiome article. Hey, we're going to do this when we actually meet together as a class. I want to hear some of your questions from the article because I'm sure you probably have some. Let's first jump to some vocabulary and key concepts. First vocabulary is bacteria. I've said bacteria before. You've probably heard the word bacteria before. But what do we mean? What do we mean when we say bacteria? Tiny organisms that are made of a single cell. Now, here's the thing. Are there other organisms that are made of a single cell that are tiny, that are not bacteria? Yes, there are. But for our purposes right now, that definition is going to be fine. We need to know that bacteria are organisms. We need to know that they're single-celled organisms, and we need to know that they are tiny. If we looked at a scale of the universe and the scale tool on Amplify, we saw that, yeah, bacteria, they can be smaller than our own cells, okay? Here is one key concept for us right here. The human microbiome contains approximately 100 trillion microorganisms, most of which are bacteria. Okay, interesting. That's a lot of organisms. 100 trillion microorganisms, and most of them are bacteria. Okay, in my experience, bacteria, I thought they were kind of bad. Is this bad for us? Does that mean that we're unhealthy? Well, let's find out. Let's explore this. Another key concept number 6, the human body provides an environment, food, and space for bacteria to survive. So bacteria like living in you because you provide them a nice, warm, wet, dark, food-filled home. And they thrive inside of you. Okay, again, this question, well, is that bad that they really like living in me? I mean, I'm kind of getting the willies right now just thinking about all these little creepy crawlies and all over me. I kind of want to go take a bath from the inside out, right? Well, hang on. Let's go back to our unit question. Remember, we asked this question: how can having 100 trillion microorganisms on and in the human body keep us healthy? Okay, so even though I'm a little creeped out, there's still this idea that maybe they're keeping me healthy. So I'm willing to continue reading on and finding out more about these bacteria before I go do my cleanse, right? Hey, look, we have another video message from Dr. Mara, the head scientist of the Microbiome Research Institute, about a more specific problem to investigate. Cool. Hey, I uploaded the video to your Google Classroom. It's called The Bill. Go watch that right now. Guess what? Guess who's going to make an appearance? You guessed it, Robert Naismith. So let's find out what that guy's up to. Go ahead, click pause and watch that video just now. Yeah, you heard it right. Fecal transplants. And yeah, that is what they are. You take poop out of one person and you put it into another person. And that's supposed to make them feel better. Well, it's a little more complex than that, but essentially, that's the idea. So, Robert Naismith, what a character. I love this guy. What questions do you right now have about the ideas presented in the video? I know I got questions. I got all sorts of questions about fecal transplants. I don't know about you. And what do you hope to learn about these topics? Now, we don't have partners with us right now, but this will be a question that we come back to and discuss a little bit as we get into our class discussion time when we meet together. Let's talk about this Robert Naismith guy. Is Robert Naismith a believable and accurate source of information? He's a politician. Shoot, he's a senator. He was elected. But is he believable? Is he an accurate source of information? Who would we want to trust in this situation, Dr. Naismith or the person who devotes their entire career towards studying this? I don't know. That's up to you. Do you think he might be biased? Why would he be biased about this? Why would Dr. Mara be biased about this? Why would anyone be biased about this? Anyways, interesting questions to think about. What do we want to do? We want to follow the data. That's what we want to do. And that's what we're hoping to base our conclusions on. So let's look at this. This is our Chapter 2 question. How can fecal transplants cure patients infected with harmful bacteria? Because, if you're like me, I'm thinking, wait, put poop into somebody? Who came up with this idea in the first place? Let's explore this, though. Let's find out a little bit. So here we go. We're going to look at patient 23. Guess what? Patient 23 ain't feeling too good. And guess what patient 23's treatment is going to include? A fecal transplant. So let's talk about this a little bit. Oh, Dr. Mara has sent a follow-up message with information about a patient. The Institute has studied this patient was cured. Oh, whoa, cured? Nice. With a fecal transplant procedure. Let's look at this message from Dr. Mara. I'm going to go full screen so we can read it here. And it says, loading. Here we go. 2 student researchers, that's you, from Mara, head scientist, subject case study of a fecal transplant patient. At the Microbiome Research Institute, we collect data from many patients that have undergone a fecal transplant procedure. We collect information about these patients so we can more clearly understand what is going on in their gut microbiomes before and after the procedure. This practice of carefully collecting data about one patient is called a case study. Okay, I'm sending you a case study about patient 23, a 64-year-old man. We collected data about this patient's gut microbiome over 9 weeks while he was undergoing a fecal transplant procedure. At the end of the 9 weeks, he was cured of a potentially deadly infection. How did the fecal transplant procedure cure this patient of a very harmful infection? Please analyze the information collected about patient 23's gut microbiome to help us answer this question as we build our argument against the senator. Geez, reminds me of like Star Wars or something. The Senator Palpatine or something. Anyways, what are we doing? We're trying to find out information. We're trying to see what can we learn about this fecal transplant procedure against the senator? Let's just sounds so antagonistic. What if we try to work with the senator? We want to inform and educate the senator, and hopefully not be against the senator, but eventually get the senator on our side in this. Anyways. Here's our patient, patient 23, sex male, age 64, data collection period 9 weeks, outcome cured. Fantastic. For a case study, let's talk about that really quick. Scientists collect data about one example over a period of time. Focusing on one case can help explain why similar things happen to other people. So we study this because we might be able to find out information that we can relate to other people and what they're undergoing. We never want to just put, all of our eggs in one basket. We never would want to just say, well, it worked for this person, so it must work for everybody. However, it can shine light and lead the direction of our research. If we do find that, okay, it actually worked for this person. Maybe it worked for this person, and we keep studying it, and we find out, well, it didn't work for anyone else. Must not be a cure, Maybe we just lucked out. But that's why we keep studying it, right? To make sure that it's actually something that works. So information about the natural world is called data. Scientists collect and analyze data to help answer their questions. We'll analyze the data collected about patient 23's gut bacteria during the first few weeks of the case study. The data is displayed in a pie chart. Let's learn how to read it. How many of you heard of a pie chart? Most delicious chart out there, but not this one, because this one's talking about gut bacteria. Hey, look at this. Patient 23's gut bacteria. Okay. Let me present it so we can see it a little bit bigger here. All right, and you have this picture in the Amplify, like that. All right, so this, in week 1, this is showing us, okay, look at this. The patient feels normal. Well, why is the patient at the doctor? Don't ask questions. So the patient is feeling normal right here, and we can see, okay, all these colors match up with certain different bacteria that are in their gut microbiome. And ooh, that is a bad picture. You do not want to see that picture when you're reading your chart. Patient reports vomiting, diarrhea, and a fever. That sounds unpleasant. I don't know about you. I don't like those things. And look, do we notice any differences between the two charts, between the composition of the patient's gut microbiome in week 1 versus the composition of the gut microbiome in week 2? What all those differences? Well, guess what? That's what you're supposed to figure out. Now in Amplify, I don't believe it gives you like an actual place to write out your notes. I could be wrong. I hope I'm wrong. Oh no, it does. Look at this. I was mistaken. After your discussion, record your ideas about why. So tell me why. What do you think is going on? And what is the evidence that supports that? There you go. Thank you. Go ahead, take a second to do that. What do you think is going on? What is the evidence? And come back to me. Hopefully, some of the differences between week 1 and week 3 were obvious. Hopefully. If you're not able to see colors very well, then maybe it wasn't so obvious. But we want to look really at this, like what's the difference and what's the same from week 1 to week 3? Okay, and that's what we really wanted to note there. Hey, next thing we're doing is activity 4. I'm going to go to number 4 in Amplify. A second read of the human microbiome. A second read? What? I already read it, Mr. Wiggin. Okay, well, let's talk about that. Scientists often use information from text to help them make sense of data. We'll return to this article because it might have information to help answer our questions about patient 23. Okay, so the first time I read it, I was just learning about the human microbiome. But now I have this problem. I'm thinking about patient 23. And now I want to go back and read it kind of with a different view, like the lens of how is this related to patient 23? What about the bacteria that I saw in the pie charts? Is there anything that I can take away from that? Am I giving it away? Oh, look at this. Sophisticated readers. Don't you want to be sophisticated? Reread texts to better understand difficult ideas. Reading more than once also builds something called persistence. Persistence means that you go back to something multiple times to better understand it. So today, as we read, we will focus on understanding why patient 23 felt normal during week 1, but sick during week 3, and what that may have to do with his gut microbiome. So here's the investigation question. How is a healthy gut microbiome different from an unhealthy gut microbiome? So check out, here's what you do. You read through it, and look, in Amplify, there's questions on the side. Oh, I love it. Great. So there's these questions. What do bacteria do in a healthy gut microbiome? So these are focused questions to help you focus your reading. So please reread this, answer these questions. Think about patient 23 because his life is in your hands, dude. So anyways, yeah, do that, reread, and come back to me when you're done. Is there any information in the article that helps you understand what's going on with patient 23? Well, the answer is yes. If you said no, I don't know, maybe go back and read it again. But we'll talk about that in our next class. Reflection time, activity 5. Yay, let's reflect. Let's talk about our reflections. So what we're going to be doing here, Now you will add to or revise the explanations that you wrote about why patient 23 felt normal during week 1 but sick during week 3. Remember when we looked at those pie charts? We said, oh yeah, look at what's going on. Well, we're going to go back to that based on what we read in the article. So think about the article, think about what you read, and then revise why you think patient 23 felt fine during week 1 and sick during week 3. If you have the same answer, that's fine. Just make sure you cite the article and what parts of the article support your answer. Okay? Bye. That's it. See you next time.